first thing I'd like to do before I talk to you is say thank you. It's with your support that I stand here before you, and I've been able to make the incredible difference that I have in the past sort of 8 or so years. So thank you very much. Your support means everything to my career. And I'm going to start off the talk by telling you a little bit about the research that you're actually funding me to do, and a little bit about the challenges that we face in this country with funding, and tell you exactly why your support through the National Breast Cancer Foundation is so incredibly important. So I'm going to talk to you about cancer. Cancer, and breast cancer, is a scary word, okay? But it's not just one disease, it's multiple diseases. And over the past 20 or 30 years, we've learnt a lot about cancer. There's still more to be learnt, but what I should tell you is that cancer is actually a disease of our normal cells. Cells actually make lots of mistakes. They have to divide all the time. There's 30,000 protein-coating genes in the body, and cancer cells have to replicate that every time they develop. Okay? Now, what are the chances of you or I making a mistake? If we had to do 30,000 tasks every day, we'd probably make a lot of mistakes. And you know what? We're really good at repairing those mistakes. But for cancer cells to actually develop, they have to acquire a number of traits. Firstly, they have to acquire the mistakes. Okay? And those mistakes give cancer cells the ability to grow, form tumours. Because they grow large, they have to also be able to recruit blood vessels to give themselves the oxygen to continue to grow. They have to also avoid the body's defences. There's a revolution going on at the moment in cancer research where we're understanding the ways in which our own immune system first tries to attack the cancer cell, but then the cancer cells learn how to use the immune system to spread and grow in distant parts of the body. They have to be able to divide forever. We don't keep dividing forever. We get to a certain height and we stop. But cancer cells keep going forever. They have to escape into the bloodstream and go to other organs. And they have to make energy so that they can do that, all of those processes. But very importantly, no matter where the cancer is in your body, the first part of the tumour or in the bloodstream or at the distant organs, they have to avoid death. Because normally, when we have a diseased cell, we have these mechanisms inside our normal cells that tell that cell to die because I'm diseased, I must die. But cancer cells learn how to avoid that cell death. And that is what you are funding me to learn about, and how I can flick the switch between life and death. So, on my 2008 training fellowship, after my PhD that I was awarded by the National Breast Cancer Foundation, I was sent down to the Walter and Eliza Hall Institute at Melbourne to really learn how cancers avoid cell death. There's some experts down there that had some pharmaceuticals that were actually currently in clinical trials for blood-borne cancers, that is leukemia and lymphoma. And those drugs were designed to flick that life and death switch. And our big question when I went down to Melbourne is I said, well, okay, if we've got a therapy that works in blood cancers, what about I use that in breast cancer and see if that works? Okay, and what I'm showing you here are 4 pictures. We've got these fantastic models where we can actually take real patient tumours, grow them in mice, and then ask what therapies work on those tumours, and hopefully personalised medicine for each and every patient. You can see on the left-hand side, all those blue dots are cancer cells. They're rapidly dividing, they're forming tumours. The next picture is a tumour treated with docetaxel, a very common chemotherapeutic, cytotoxic chemotherapeutic that works pretty well. Those brown cells are the dying cells. So we treat the tumour with the docetaxel and we get some cell death. So we start to kill the tumour off. When we use that novel chemotherapeutic on the same tumour, we got some death, but not a lot. But the really profound discovery that we made when I was at Melbourne is that when we combine those 2 drugs, now look how many brown cells there were. And what's the amazing thing is these pictures were taken 24 hours after I started the therapy in these mice, and at 8 days, the combined therapy, the tumour was gone. And so that was really amazing. Now that sounds fantastic, but we worked out that there's about 10% of triple-negative breast cancers that would respond to that. And the cool stuff is that in those tumours, we can almost cure the mouse of disease, so cure patients of that disease. you can see the combination treatment with that protein, that sort of treatment that turns the cell death back on. You can see the standard chemotherapy in blue. You can see the tumour responded initially and shrunk, but then acquired resistance. Just like breast cancer patients acquire resistance to certain therapies, that tumour started to grow again. But when we turn that cell death protein back on, you can see that the tumour never came back. So that was really great. And that therapy now is in a clinical trial for breast cancer. So that's cool. We've made a big difference on that. And it's actually funded by the National Breast Cancer Foundation from my old colleagues at WeHi. So now, there's also multiple ways in which cancer cells survive. And now the research that I'm funded by National Breast Cancer Foundation at the Garvan Institute to do is I'm working on a different cell survival protein. There's many. And I'm trying to learn all the different ones and how I can switch them off. This one I'm working on at the moment is a survival protein. It's there in every cell in your body and it tells cells to live. I'm strong and I live. And I've learned a way how to turn that protein off. Now this is a survival curve. It's a technical kind of diagram that us scientists look all the time. The black line there are all the mice that are just controlled, not treated. And all those dots are when the tumours reach the ethical endpoint and we've said, okay, that experiment is ended. When we turn that survival protein off, we more than double the survival time of those mice. In fact, I've got mice that are on this therapy at the moment that I might have to take away because they're actually reaching the end of their life. Wouldn't that be cool that we live with breast cancer as a chronic disease? So that, it's there, we just treat it and, we probably get old before it actually makes, any difference to our body. So that's what I'm trying to achieve. Whether we can get rid of the cancer altogether, don't know. But if we can make it a chronic disease, something that we can manage, that would be even wonderful. And this is really cool. When I turn that survival protein off, the other thing we discovered recently, and these fancy diagrams, but all those yellow dots, they're cells. They're like the brain of the cell, right? And all those yellow dots are where we see damage to the DNA. And these cells have actually been treated with irradiation. Irradiation is obviously used in quite a number of patients to actually irradiate the bed of the tumour to try to get rid of any residual cells. And what irradiation does is it damages the cell and kills it. But sometimes cells don't respond. Interestingly, what we've learned is that when we turn this survival protein off, this same protein I was showing before, there are more dots. When it's on, less dots. When it's off, we get more dots, which means the cells are more damaged and they'll die. So we're actually making a real difference on the cancer cells by turning this survival protein off. And this is the really cool thing. This is the latest things that we've discovered. We discovered this year. This is a survival protein. It's the switch between life and death. What we've actually recently discovered that it doesn't just regulate that switch between life and death, it also regulates the way that cell gets through normal tissue. And this is a lab-based assay, but basically all the blue stuff and all that sort of lighter blue stuff is what your breast would be without the cancer cells. It's kind of like the tissue, the collagen and everything like that. The brown cells are the cancer cells. On the left-hand side, they're untreated. And the cells get through and they have these mechanisms that they actually erode that collagen tissue to get through and spread to other organs. We've discovered that when we turn that cell survival protein off, they're not able to do that as well. And so we're also discovering that that protein's involved in invasion. So we started asking, if that's the case, then can we stop the cells from getting to the lung, metastasizing? And this is what we've done here. All the top panels there are the cancer cells. The brown are the cancer cells. We've stained them to look brown cancer cells. And you can see cancer cells there in the control, cancer cells when we've turned it off. The tumours grow slower, so we already know that. And on the right-hand panels are the lung. You can see the holes, that's where we breathe through and that's where we get oxygen. And those brown cells are again where the tumour has spread to the lung, so the metastasis. And down here, we're actually doing a stain for death protein. What we've discovered is that when we turn off our therapeutic, the cells are unable to get to the lung. So that cell survival protein not only is involved in life and death decisions, it's involved in invasion, and we can actually prevent those cells from getting to distant organs. Because ultimately we've cured primary breast cancer. We do it with surgery. We take out the primary tumour, but why patients die? It's because those cells get to other places, and that's what we're trying to discover within National Breast Cancer Foundation Fellowship that I'm funded by. So this is my latest work, and I'm hoping to publish it. You know, we're doing some really great work. I'm also funded by the National Breast Cancer Foundation to have a research assistant and also a PhD student who's doing this fantastic work. We hope to publish it and really make a difference, because guess what? My collaborators at WeHi are starting to make the pharmaceutical against this protein, and if I publish my research, that may expedite that particular pharmaceutical into clinical trials. So it's very exciting. Now, that's my research. Quick tell you about the state of research funding in Australia. This is why you guys are here. You think, oh, well, the government funds research, there's $400 million, they give us $400 million every year for medical research. That's not just cancer research, that's not just breast cancer research, that's all medical research. Dementia, heart disease, everything's very important, but it's only $400 million. You think about how much the government spends on other things. It's not very much money when we're punching well above our weight in Australia for medical research discoveries. And this is the state of funding in Australia from the National Health and Medical Research Council. Last year, there were 400 applications that went into the National Health and Medical Research Council, and in 2014, they only funded 14.7%. Okay, you think, oh, well, maybe some applications weren't so good. Well, this is the data from 2003. Don't have to read the data, but basically the green ones are the unfundable ones. The ones that us scientists deemed as, well, it might have made a difference, but you know what? Probably not there yet, probably not going to make a difference in patients. The blue and the pink are all the applications that went into the National Health and Medical Research Council that we went, you know what? That's going to make a difference. Someone's going to survive because of that research, but the pink are the only ones that got actually funded. There wasn't enough money to fund all the blue. So what does National Breast Cancer Foundation do? They pick up some of the applications that didn't get up into the pink zone. They're not bad applications. They're still really good applications that could make a real difference in the clinic. But there's just not enough money to go around. We're really smart people in Australia. We have a lot of really smart people. That's why there's 4,000 applications every year. But there's just not enough money to go around. So your dollars make a big difference in enabling me to be a scientist here standing before you today to make those fantastic discoveries about metastasis and cell survival. That's what you guys do. So I want to thank you for that. And I'm not even going to touch on this for too long, but if we think about women in science, it's even more dire. Because of the funding crisis in Australia, and because women have to take time out for babies, like I did, it's really difficult to get back in the lab and actually get those applications up. So you can see in the more senior positions, there are very few women surviving in senior research positions. And your research dollars fund my early career research fellowship that I may not have actually got from the NHMRC because of your dollars. So again, thank you. Now, and I'm sorry, Amanda, I know you're here, but I've been going to Amanda's Pink Ribbon Breakfast for the last 3 years, and it's been an absolute pleasure to be there. Why do I do this? It's because, one, if I can live my life and my career knowing that I've actually made a difference in someone's life, I've made a mother, a sister, or even a father for the rare men that actually develop breast cancer as well, that I've improved their lives, then I've lived a good life. And I get the pleasure of going to breakfasts like the one Amanda puts on every year. And, you know, you're just awesome people. I thank you so much for your support for my research and everyone's research in Australia. It's so important for us, and it means that I get to work every day and I can make that difference. So each October, your Pink Ribbon Breakfast raise important funding for us to actually do this life-changing research. Please keep up the good work, and thank you again. And why do I do this? Well, you know what? I've been talking about breast cancer, but I told you about a pharmaceutical that I took from the blood system, from blood cancers, and I made sure that it worked in a breast cancer. You know what? We can do the same thing from the stuff that I discovered from breast cancer, and we can actually maybe translate that into other cancers as well. And I put this picture up because this is a little girl that I know and good family friend. And she was actually given the wonderful news this year after being diagnosed with acute myeloid leukemia that she was cured. And it's because of life-saving research that we can do that. And I want to do that for each and every one, breast cancer patient or cancers alike. If I can make a difference in my life and more people like this beautiful little girl can get the cure when they walk into the doctor's office, then I've done a good thing. So thank you very much for your attention, and it's a pleasure talking to you.